Today we will be beginning Chapter 10, covering blood. Now, so blood, when we talk about blood, it's the only fluid tissue in the body. It is classified as a connective tissue, and it has two main components. We have the living cells, the formed elements. We'll talk about those in detail today. And then the non-living matrix, the plasma. So just to recall that with our connective tissues, every one of them is going to have our living cells, and then they produce a matrix that's sent outside of the cell. So if blood is centrifuged, now what that means is we take it in a machine and we spin it really, really, really fast. Now, my other job at the hospital, I use these on a daily basis. Now, if blood is centrifuged, erythrocytes, our red blood cells, will sink to the bottom. They have more mass. Now, by volume, that should be about 45% of your blood. It can fluctuate a little, but it should be about 45%. And this percentage is known as the hematocrit. Now, the buffy coat is going to contain leukocytes, your white blood cells, and your platelets. And that's going to be a very small percentage, usually less than 1%. It's really thin, this whitish layer between the erythrocytes and plasma. And then plasma will rise to the top. It's the least dense, and it will make up the majority of the volume at 55%. So I figured 10.1 shows essentially what happens when we spin it in a centrifuge. So your plasma, now it's primarily going to be water and ions, and then also your substances that are transported by blood. So your nutrients, your waste products, all of that as well. Now, in your formed elements, your erythrocytes, okay, you have your red blood cells. In that buffy coat, you have your white blood cells, your leukocytes, so things like basophils, eosinophils, neutrophils, lymphocytes, monocytes, all that. These all deal with immunity, okay? And we'll talk in more detail about those in just a few minutes. And then lastly, your platelets. Platelets are involved in blood clotting. We'll talk about their importance when we get to hemostasis later. Now, in terms of the physical characteristics of blood, your color range, it should be scarlet red to a dull red. Now, one thing that I find interesting is when blood is deoxygenated and we see these blue veins in our skin, blood is still red, but it's just that our skin gives the bluish green appearance of veins and not the blood itself. So pH, we need to maintain relatively constantly 7.35 to 7.45. Outside of this, we get into blood acidosis or alkalosis. It's not good. We need to maintain that pH in pretty tight range. And it's slightly higher than body temperature, about 104 Fahrenheit, about 38 degrees Celsius. So in a healthy adult, blood volume is 5 to 6 liters, or about 6 quarts, and actually makes up 8% of body weight. Okay, so that's quite a bit. Now, again, these values right here are average. Remember that this is homeostasis. It's dynamic. Okay, so sometimes you may have less blood, sometimes you may have more. It may, if you are above or below average body mass, it could be more or less than 8%. The temperature can fluctuate, pH can fluctuate, all of that. So remember, it's dynamic. These are simply the averages. So let's talk about plasma now. It's approximately 90% water. Again, we saw in figure 10.2, carries as nutrients, your salts, respiratory gases, hormones, plasma proteins, waste products, all the stuff that we needed to transport in our blood, we classify it within the plasma. Okay, so plasma proteins are the most abundant solutes or particles that are dissolved in there. Most of them are made by the liver, things like albumin, clotting proteins, antibodies. Okay, so your clotting proteins help to deal with forming or stopping blood loss. So if you have a laceration or scrape or whatever the case is, your antibodies to help protect the body from antigens. So we would say if you got the COVID vaccine, those antibodies that your body produces, they're circulating in the blood. That's considered part of the blood plasma. Okay, so I talked a minute about acidosis and alkalosis. So acidosis occurs when blood becomes too acidic, when we drop below that 7.35 average, and alkalosis occurs when we become too basic, we move above that 7.45. So in each scenario, the respiratory system and the kidneys help to restore this blood pH to normal. So if we have too much CO2, the respiratory system will aid in removing that excess CO2. The kidneys will help to trigger the production of more blood, extra filtering of blood to get any substance that may cause alkalosis or acidosis to occur. Okay, so your formed elements, we've already talked about these. The last one, those cell fragments. So we do break our red blood cells down. We'll talk about that in just a minute as well. Okay, so this is what is called a peripheral blood smear, photomicrograph. So if you work in a hospital lab like I do, I've seen tons of these. So we take a drop of red blood, and we put it onto a microscope slide, and then we literally smear it across the slide, and this is what we do. And if you work in hematology, that's the study of blood, you will actually take this normal, smear right here, this view, this would be under a microscope, and you count these. Okay, this is what's called a peripheral smear manual differentiation. So we go in and we count all of these here. And as you can see, the most abundant one that we have here are these disc-like erythrocytes, red blood cells. Now, we also have our other formed elements that we see right here. These tiny dots are going to be platelets. We have lymphocytes, neutrophils. You could also have eosinophils, basophils, other ones as well. Okay, so table 10.2 is a great table. You're definitely going to want to use this for your assignment. It gives your basic information on all of your different formed elements. Okay, so the erythrocytes, their main function is to carry oxygen, and they are biconcave disc. So they're kind of like a donut, only the middle isn't all the way cut out. They're really just bags of hemoglobin. This hemoglobin is what carries the oxygen. They are anucleate. Okay, they have no nucleus. Now, because they have no nucleus, they cannot go through mitosis. They can't replicate themselves. Now, that's actually, a pretty good thing. The reason being, we've kind of talked about this idea of maintaining homeostasis at the cellular level. So if you have a cell that's highly susceptible to damage, like the skin, for example, we talked about the epidermis, that those cells are keratinized, they die, and then they simply fall off. Okay, our red blood cells, they're the same way. They have a literal shelf life of about 8 weeks. Okay, we'll talk about the process of red blood cell production in our next lesson, but it's pretty interesting as to why they do that. Okay, so hemoglobin, it's a protein that contains iron. It binds very strongly but reversibly to oxygen, and it allows us to be able to carry oxygen more efficiently in our blood. So every red blood cell is quite literally just covered in hemoglobin molecules. They have about 250 million, guys. It's 250 million hemoglobin molecules, and they all aid in functioning to carry oxygen within the blood. So if our red blood cells, if we get imbalanced, okay, if you have anemia, this is a decrease in the oxygen carrying capacity of blood. With anemia, you can have things like lightheadedness. If you stand up too quickly, you get everything kind of fades to dark. So that indicates that you need to maybe take an iron supplement or maybe eat some red meat. Okay, sickle cell anemia. This results from abnormally shaped hemoglobin. It's hereditary. It's genetic. There's a genetic base for it, and we'll see in a second what the blood cells look like. And then polycythemia is when you have an abnormal number of erythrocytes. You have either too many or too low, or not enough red blood cells, and having too many can be just as much of a problem as not having enough. Okay, so table 10.1 goes through the different types of anemia. You can read through those. Again, you'll need that for your assignment today. Now, we can see right here in figure 10.3, we have on the left normal hemoglobin, this this nice regular shaped erythrocyte right here. And then on the right, we have this sickle, or this crescent shaped cell. And it's because of this single base change, what's called a single nucleotide polymorphism, or a single nucleotide change, causing the entire red blood cell to change shape. Okay, so let's briefly talk about our leukocytes, our white blood cells. They're crucial in the immune system. Some of them can move by amoeboid movement. They can move into and out of blood vessels as well. They can respond to chemicals released by damaged tissues. They need to do this so that they can go to those damaged tissues and either repair them or get them out of the body. And there's a much, much lower count by volume per cubic millimeter than erythrocytes. So we see 4 to 11,000 right here. If we jump back, we see was under 5 million. Okay, so a much, much smaller proportion in comparison. Now, if we have an abnormal number of leukocytes, so again, let's imagine you're working in a hospital lab in the hematology department. So if you have leukocytosis, this is when you have a white blood cell count above 11,000 leukocytes per cubic millimeter. Now, cubic millimeter, that's the volume that's going to be in one of those little tubes that they draw at the doctor. Now, this is generally going to indicate that you have some type of infection, that the immune system has been activated, and you are trying to, make sure that your body has triggered that to try and figure out what's wrong with the body and get it corrected. Now, with leukopenia, this is an abnormally low leukocyte level. You could have something causing, an issue in the production of white blood cells. Certain drugs like corticosteroids that you're prescribed when you're sick can do this. Anti-cancer agents as well, so they can drastically decrease your white blood cell count as well. And also drugs that are used after a transplant as well. And that's actually done so that the body will not attack the organ that's been transplanted. So leukemia, this is bone marrow becomes cancerous. It turns out excess white blood cells. They then go on to usually form tumors throughout the body, causing other cancers as well. So in terms of our leukocytes, we have granulocytes and agranulocytes. Granulocytes have granules, which can be stained. It's going to be our neutrophils, eosinophils, and basophils, and our agranulocytes do not. Their nuclei are spherical, and they include our lymphocytes and monocytes. Okay, so just an easy way to remember those are white blood cells from most to least abundant neutrophils, lymphocytes, monocytes, eosinophils, and basophils. Okay, so figure 10.4 shows how they arise. So they all come from hemocytoblast stem cells, and they branch into either lymphoid stem cells, giving rise to the lymphocytes, or myeloid stem cells, which give rise to all of the other formed elements, including our red blood cells. Okay, so with our granulocytes, our neutrophils, these are multi-lobed. They act as phagocytes at active sites of infection. So if you have, let's say, a pretty deep cut, you went to the doctor, had to get stitches, and maybe that cut didn't get cleaned out all the way. Neutrophils will go in and try to get rid of any infection that's there. Now, eosinophils are found in response to allergies and parasitic worms. So when there is a foreign contaminant within the body, eosinophils will be activated to try and remove it. Basophils, these will initiate inflammation. So if inflammation is a body response to try and prevent something from getting into the body or something from spreading. Okay, so when you get bitten by a mosquito, that inflammation that occurs is to help prevent that toxin that the mosquito has put into you from spreading to other parts of the body. So lymphocytes play an important role in the immune system. They will help to break down foreign substances. Monocytes function as macrophages. They will quite literally ingest foreign particles, and as they do so, they will help to fight infection, bring in things that shouldn't be in the blood. Okay, so platelets. Now, platelets are needed for the clotting process. So if you have a some type of cut, laceration, and you need to stop that from bleeding, we have our platelets come in, and they form a temporary patch, so to say, to help prevent us from bleeding to death. Okay, so there are some situations in which you have an abnormal issue with platelets. So if you have no platelets at all, then you wouldn't be able to clot at all, and you could bleed to death from a simple paper cut. So it's very important to have these platelets, and we'll talk about their role in more detail in our next lesson, hemostasis. All right, so if you have any lessons, put it in the comments below. You can send an email to mr.tucker.schs@gmail.com.